Hello guys, welcome back to our channel Biolab India. This is Vamsi, and in today's video we will be completing biotechnology principles and processes. Yes, I have missed. Yes, yeah, processes in one shot. Okay. The thing is, no need to get your notes anything, but listen this video or listen this lecture with much of focus on it. Put much attention on this lecture, memorize the topics within the lecture itself, then go back, open your notes and like not your notes, sorry, the NCERT textbook. Open the NCERT textbook, give one reading. I am promising you that I will not try to miss even a single point. I will not try to miss. I will be covering all the points here. If not, if there is one or two points that is left, you cover it while you are reading your NCERT. Give one reading, it is enough. No need to write notes as anything. Just memorize the concepts in this video only. This chapter is so easy. This is so easy. No need to worry much about this chapter. Okay, this is so easy. I'll make it easy for you. Let us move on with today's video. Yes, starting with biotechnology and principles. Let us start with the question: what is biotechnology? Yes, what is biotechnology? In order to understand this, our NCERT has given three different phases of biotechnology. One, it is the old phase, one, it is the present phase, and the third one is the definition which is given by European Federation of Biotechnology. These are the three things which are mentioned in our NCERT in order to understand this word biotechnology. Now let us starting with the old definition of biotechnology. That, see, have you ever seen your mother preparing curd from milk? Yes. Do you know about wine? Yes. Since we have completed that in the microbes and in human welfare chapter. Yes, we have completed that in the microbes in human welfare chapter. So if anyone haven't seen that, go there. There is much detailed explanation how wine is made, how bread is made, how curd is made, like that. There is a detailed explanation on it. Go watch it. Okay, those were the first initial days when I started this chapter. Sorry, in this channel. That was my first you know first chapter which I have covered. Okay, so we are preparing curd from milk using microbes. What are microbes? Living organisms, right? Microbes are living organisms. So therefore, once upon a time, yes, old age biotechnology. Let's try with this. Old age biotechnology meant, in the olden days, the biotechnology meant that, see, living organisms, yes, live organisms, and enzymes. We made the things that are necessary for human use. Yes, we made the things that are necessary for human use. See, we have used milk, we have made curd. Curd is necessary for our human use. Even milk is also necessary, but curd is also necessary, right? From we are making this curd from milk using microbes. So in the olden days, we have used these live organisms or enzymes. Yes, or enzymes, any enzyme we have used to make products and processes, to make products and processes that are useful to humans. Clear clear. And these techniques, yeah, that's not visible, right? And these techniques which were used to convert which were used in order to convert this milk to curd using those live organisms which are useful for the human welfare, which is useful for human welfare. These techniques were known as biotechnology. These techniques are known as biotechnology. This was the definition, this was the old age definition of biotechnology, okay? Of biotech. This was the old definition of biotechnology which was given in our NCERT in the initial lines. Okay, now we are new people, right? Our present generation is a new people, right? So we have to modify this definition. We have modified this definition into our own definition, that is your new age definition of biotechnology. What is the examples for this? Making of milk from curd, making of bread, making of wine. These were microbe mediated, okay? If yeah, here we have space, right? Write it here. Curd, bread, wine, etc. Wine, curd, bread, wine, these were used as microbe mediated. These were microbe mediated, okay? Yes, bread and wine are made from yeast, fungus, unicellular fungus. Yes, that is a microbe. So these are microbe initiated. This is the definition as of biotechnology as per the olden days. Now let us see the definition of biology in the present era. Seeing the definition of biotechnology in the present era. Have you ever heard about genetically modified organisms? Yes, sir. Yeah, wait a minute. Organisms. Yes, GMOs, genetically modified organisms. Have you ever heard about them? Yes. Now, using these genetically modified organisms, we are producing many things on a large scale. Using this genetically modified organisms. What are genetically modified organisms? The organisms in which their genes have been modified for human purpose. The genes of these organisms have been modified for human purpose. We have seen such examples in the the microbes in human welfare chapter. This and that chapter is a little having a little correlation between them. Okay, these are genetically modified organisms. These genetically modified organisms are used on large scale in order to produce products that are useful for human welfare. Everything is for human welfare, guys. Everything, every single thing that we do on this earth is for our welfare only, but not for the welfare of any other organism. Okay, on large scale, they are used on large scale, and products are produced, and this is the new definition. Okay, write it here: present biotechnology. These processes lead to biotech or biotechnology. Okay, clear, clear. Now the thing is, what are these processes that we are doing as an example of biotechnology? Now, you said curd from milk using microbes. We don't want that. We want something that has to be cut into pieces, something that has to be put, something that has to be joined. Yes, yes, yes. Wait a minute. If I try to decrease the size of this, this is fine, right? A little fine. Yes, this one is good. Now, the biotechnology, you have to cut it, you have to put the gene, you have to insert it, you have to paste it. Everything should happen. Where are those? That is why we are here to learn biotechnology. You are not telling us that. You are just telling microbes are used. Curd is produced from milk. No, that is this definition. So, the examples of the present day biotechnology are in vitro fertilization. Yeah, now we are telling us the original biotechnology. Yes. In vitro fertilization. Clear? In vitro fertilization. This in vitro fertilization is only called as test tube baby. Test tube baby. Clear? Yes. Next one. Gene synthesizing. We are synthesizing new genes. We are synthesizing new genes using biotechnology. This is the first one. This is the second one. Gene synthesis. Yeah, gene synthesis. Developing vaccines. We are developing vaccines. Like the vaccine for corona. Developing vaccines, especially DNA vaccines. DNA vaccines. Okay, do not worry about these vaccines. We will be covering it under human welfare. Human health and disease chapter. Okay, correcting defective gene. Yes, we are correcting defective gene. That is in your chapters. Yes. Oh, sorry. SCID. Yes, SCID. What is SCID? Severe immunodeficiency. SCID. SCID. Yes, SCID. The ADA deficiency disease. The disease which is caused due to the deficiency of ADA. Severe combined immunodeficiency. Okay. Not deficiency, disorder. Yes. Defective gene. What we do is we take out lymphocytes outside. In that lymphocytes we have ADA gene. That ADA gene is cut and it is replaced with the functional ADA gene, and then these lymphocytes are again cultured in larger amounts and they are again sent into the human body. This is your SCID, the first gene therapy that is done. It is done to a 4-year girl. Okay, 4-year girl. This is the present day biotechnology. Okay, these are the present day biotechnology. Now let us come to the European Federation's definition of biotechnology. Yes, European Federation of biotech. Now the thing is, the definitions which we have discussed here the people who gave these definitions were fighting that our definition is correct, that our definition is correct. Now EFB has come, European Federation of Biotechnology has come, and it has solved the dispute by saying that it has solved the dispute by saying that biotechnology involves both traditional as well as modern molecular uh, processes and techniques. It has solved the dispute. As per European Federation of Biotechnology, the definition of biotechnology is it is a combination or emphasis of both traditional. See how simple it has solved the dispute. Traditional, it hasn't ruled out any traditional process that making milk from curd is not biotechnology. No, it hasn't ruled out. Plus modern usage of genetically modified organisms is not biotechnology. It hasn't ruled out. Is equals to biotechnology. This is the definition given by European Federation. Not the definition, but the definition included these two parts, both the parts. Now the exact definition that is given by this European Federation is, see, look carefully here. After writing this definition, I'll be again starting recording, no need to waste time here. Okay, so this is the new definition of biotechnology according to the European Federation. According to the European Federation of Biotechnology. A little a minute, I will not be able, you will not be able to see me. A little a minute, okay? A little a while. See this now. The integration of natural science, okay? The integration of natural science and organisms, their cells, cell parts and molecular analogs for products and services is termed as your biotechnology. The definition given by European Federation for Biotechnology is this. Clear? Clear? Yes? Clear? Good? Are we great to go? Yeah? This is the definition that is given by the European Federation of Biotechnology. It includes both traditional, see, it includes organisms. It is including organisms as well as molecular analogs. Molecular analogs is the new age biotechnology word, and organisms is the olden age. Yeah, natural science is an olden age. Okay, using cells and parts is an olden age biotechnology, and this molecular analogs is your present day biotechnology. So this is true. This is true. Traditional biotechnology plus modern biotechnological techniques gives you biotechnology as per EFP. Okay, clear? That is about the introduction to biotechnology. Now, see, biotechnology. Have you ever seen this gene modifying anywhere? Somewhere we have seen this gene modifying. Yes, we have seen this gene modifying in your sexual reproduction. Let me say, in your sexual reproduction, variations occur. Variations occur. So, variations occur. But the thing is, difference between sexual reproduction and genetic engineering. Yes, in this genetic engineering, actually biotechnology's main principles are two. One, genetic engineering. One, it is the genetic engineering, and this sexual reproduction is also a genetic engineering. It is termed as natural genetic engineering or genetic engineering of nature. So the second principle of biotechnology is your if you are trying to experiment on any microbe, you have to maintain a sterile environment. That is your second principle of biotechnology, and this sexual reproduction is the nature's biotechnology. Why we are calling this as? Because there are, in sexual reproduction, there is one male and there is one female who are coming together in order to produce two children or children. Yes, one or two. This is your child. Say, for suppose they are having two children. Okay, they are coming together to produce one single child or two children. Now, while coming together, we have seen this in reproduction that fusion occurs. Yeah, fusion of DNA takes place. During fusion of DNA, during copying of DNA, what happens is there are some variations that occur. So, sexual reproduction leads to variations. Variations are nothing but they are the new genes. If the sequence is GAATTC, due to some variation, I'm just taking an example that is in restriction under nucleoside, I know, but I'm considering it as an example, so let's leave it AAGGCC. This was the initial thing, but while copying, what happened is at the place of the first A, there was you can say some C occurring. There is C. Yes. So CAGGCC would become the new sequence of the child. So this led to a variation. This is a completely new sequence. We have seen that changing even one letter in the sequence can change the meaning. Yes, we'll be seeing, if not in the genetics chapter. Yeah, in the genetics we have seen that changing of even one single letter would change the whole meaning of the sentence. So the same thing is happening. Okay, variation. Sexual reproduction leads to variations, and this variation is nothing but the so-called mutation. Sometimes it has occurred very drastically, and using these variations we have created hybridized animals. And these hybridized animals are the traditional techniques of biotechnology. Not only animals, both plants and animals. Okay, I have written there only animals. Traditional biotech. Traditional biotech. This is in the olden times. So what they do is, see, for suppose there is a plant. See, there is a plant. Okay, I'll be taking a new slide. Wait a minute. Where we have down space, right? There is a plant A. There is a plant B. This plant A is highly resistant to diseases, but it is giving low yield. This plant B is high yielding but low resistant to diseases. Now the olden days people what would do is they would have crossed between these two plants A and B, and they produce some plant C, and this C includes both the characteristics, that is disease resistance and high yielding variety. Both disease resistant and high yielding variety. Got it? Got my point? This C plant contained, it is resistant to diseases as well as it is high yielding. Okay, so this was the traditional hybridization method, and how they have done this gene modification and this biotechnology using sexual reproduction. Sexual reproduction is a nature's genetic engineering or nature's biotechnology, if you want to say, very in a simple meaning. Okay, but the thing is in sexual reproduction, undesirable characters are also produced. Undesirable characters also occur. Okay, in which SRP, sexual reproduction. But in genetic engineering, the thing is only desirable characters are produced. Only desirable characters This is the difference that makes from genetic engineering and sexual reproduction. You cannot choose between the characters that you want. While you are mating these two plants with a high yielding variety and more disease resistant plant, there can be some other unwanted, there can be some other unwanted or undesirable characteristics that can also appear in seed plant. This is some I'm making a tick with yellow, so that yellow tick represents those characters which are unwanted for us. But when you do genetic engineering, the thing is you have to collect the genes which is responsible for disease resistance from A, and which is responsible for high yielding in B. You have to combine them, and you have to form a new gene. You have to insert into a plant, the same species, and that gene replicates itself there using a vector. We have to introduce into the host plant using a vector, and it forms many genes of that sort which are of high yielding as well as disease resistant, and the plant becomes disease resistant and high yielding. And now when you produce many such plants, that is biotechnology. This happens only with the desirable characters. That is a technique that is used in biotechnology. You can produce the plants or animals or offsprings with your own desirable characteristics. If you want a tall plant, you can regulate the gene for tallness and you can produce a tall plant. If you want dwarf plant, you can reduce that gene which is responsible for tallness, and you can produce that dwarf plant. Okay, like that genetic engineering and biotechnology are ruling the world even today. Okay, that is your thing, that is your biotechnology. Now this is about the genetic engineering and sexual reproduction, a difference. This is the difference between genetic engineering and sexual reproduction. Now we will be seeing the features in biotechnology. Okay, the features in biotechnology and recombinant DNA, which was done by Herbert Boyer and Stanley Cohen. Now see, you wanted to produce a plant C with high yielding, okay, HYV I am writing, it should be HYV, high yielding variety, as well as disease resistant. Okay, you want these two characteristics in a plant C. How will you do this? If you put the piece of this alien DNA piece which is disease resistant from another alien organism directly into this organism, directly, okay, not indirectly but directly into, if you put directly into plant C, will this plant C accept these two? No, it will recognize that as a foreign particle and it will kill that two genes. It will kill that. This is not possible. So what do you do is you isolate a plasmid. Okay, you isolate, not plasmid, that is in bacteria, not plasmid. You isolate a gene, you isolate the DNA, you cut the DNA at the place where you want to stick and paste this gene. You have to paste this gene in the genome of that plant C, then only it will replicate. Okay, now what you do is and that procedure is called as integration, simply in a literal sense. Okay, in a literal sense, integrate. If you integrate the genome which is responsible for this disease resistance and high yielding nature into this genome of plant C, integrate this genome into the genome of plant C, then you will be successful in your project, and you can produce a plant C with both high yielding variety, it is a high yielding variety as well as disease resistant variety. Okay, now what is the site that is responsible if you put this genome into that genome of this plant C, which has helped it to multiply? Why did the plant C hasn't recognized it as a foreign material? You are putting this, you are incorporating this new gene into the genome itself. So therefore the plant C cannot recognize that as a foreign particle. It will not kill that, but rather if the gene which you are inserting, has a site called ORI, which is called as origin of replication. I'll be talking about this within in the further lecture, origin of replication in detail there. Replication, if this site is available in the gene which you have inserted, it is able to replicate in itself in the new host. It is able to replicate itself in the new host, and this method, after introducing many things, after introducing this high yielding variety gene and disease resistant variety gene, you will produce it in many quantities. Due to the presence of this ORI, many copies of that gene are produced, many copies are produced, and this process of producing many copies using this ORI or region of replication is called as cloning. Okay, let me write it with another pen. This process is called as cloning. Got it? What is cloning? That process is called as cloning. Now, before moving into the chapter, I have to tell you some basic terminologies that are used here. Okay, basic tools that are used in the recombinant DNA technology or biotechnology, that is given in your NCERT after the experiment what is done by Cohen, Stanley Cohen. But I am telling you now because it will be helpful to you there in the experiment. Okay, see, put the heading tools for biotech. The first tool is region of replication ORI. Okay, that is complete. Now, the first tool that is required for biotechnology is, there is a bird here, that's what I am saying here. It is sounding. Go. Okay, no problem. Where we are? Yeah. Restriction nucleases, restriction nucleases. These restriction nucleases are of two types: restriction endonucleases, restriction exonucleases. What are these restriction nucleases? What are these restriction enzymes? First of all, actually these restriction endonucleases or enzymes are discovered in 1963 in Escherichia coli. How they discovered these? Okay, how they discovered this? I'm telling you the discovery of these restriction enzymes. Actually, in Escherichia coli, bacteria is affected by a virus called as bacteriophage. Yes, say for suppose this is your Escherichia coli, this rectangular thing, and this pink color one is your bacteriophage. Whenever this virus sits on this bacteria, it releases its DNA into this bacteria and it produces many genes in that bacteria. Yes, many genes in this bacteria, the same genes replicate and new organisms of this virus are formed inside the bacteria. But the virus has to the bacteria has to protect itself from the virus. So now what does bacteria do is it releases two enzymes. This bacteria, in order to stop this, it releases two enzymes. There are, let us say, one is E1, next one is E2. Whenever these two enzymes are released, this E1 cuts away the viral DNA that has entered. It has recognized a foreign particle, right? Viral DNA. So it cuts away this viral DNA. Now you can ask a question that this enzyme 1 can cut the own DNA of the bacteria, right? But that doesn't happen because there is E2, enzyme 2, which adds methyl groups to the bacterial DNA in order to prevent it from cutting. Now viral DNA is cut into pieces. Now there is no chance of replication. Yes, let me see the time. Yeah, it is 3:40 now. Okay, no problem. It has no chance. Viral DNA, once it is cut, it cannot divide itself. No, it has no chance of dividing. And now they have named this enzyme 1 as a restriction enzyme. Why? Because it is restricting the disease that is coming with the viral DNA. This enzyme 1 is called as restriction enzyme. Okay, or restriction nuclease because it is cutting the nucleic acid. Nuclease is the material, or it is an enzyme that digests the nucleic acids. If it is DNA, you use DNAs. If it is RNA, you use RNAs. They will eat away this DNA and RNA respectively. Now, these restriction enzymes are of two types. I have told: restriction endonucleases, restriction exonucleases. What are restriction endonucleases? Restriction endonucleases cut the DNA. Inside in the middle of the DNA. See this. If this is some DNA, say for suppose, and if the restriction enzyme cuts here, it is called as restriction endonuclease. If that restriction endonuclease cuts here outside, it is called as restriction exonuclease. Restriction endonuclease, restriction exonuclease. Until now, 900 restriction enzymes REs have been isolated from 230 strains of bacteria. Okay, the first restriction endonuclease or restriction enzyme is HIND2, Hemophilia influenzae 2. And one of the best and famous examples for this is your EcoR1, which we'll be using very frequently in this chapter. Okay. EcoR1. Let me explain you this nomenclature. Right here, EcoR1. EcoR1. E means genus of that organism, genus. Co means species. R means strain. I or 1 means it is the order in which that has been recognized. It is written in Roman numerals. Okay, EcoR1. Eco, E means genus. What is the genus of Escherichia coli? Escherichia. Yes, this EcoR1 is isolated from Escherichia coli. So Escherichia coli, genus from which this restriction endonuclease has been isolated, genus of that organism. So what is that? Escherichia. Co means coli, the species of that organism from which this has been isolated. R means strain of that bacteria from which this has been isolated. It is generally Escherichia coli strain are 1. 1 means it is the order. It was the first restriction endonuclease which was discovered in this order. Like the same way, try for Hindu 2. See, H means genus, IN means species, D means strain, 2 means order. Now what is the genus? This has been isolated from Haemophilia influenzae. H means genus, Haemophilia. IN means influenza, species. D means strain. 2 means it is the second one which is discovered in this order, but it was the first one to be discovered in whole restriction enzymes, restriction endonucleases. Okay, this is about restriction endonuclease. Now coming into another one that is called as plasmid. What is this plasmid? Okay, now we are coming into plasmid. See, plasmid is, first of all, it is an extra DNA which is present in bacteria. Okay, bacterial cells mainly. Plasmids are found in bacterial cells. They are circular in shape. Okay, circular in shape. These are double-stranded only. Double-stranded DNAs which are circular, which are found in bacterials. Generally they are extracellular. Like there is the bacterial cell DNA is there. The genome of the bacteria is concentrated at the center. It is there. But in excess to this DNA which is present in the center, there is other DNA in these bacterial cells. Why is this always coming? I'm getting irritated because of this. Okay, there is an extracellular circular DNA that is called as plasmid, and we use these plasmids as vectors in many cases. What are vectors? Yes, vectors or cloning vectors. Name cloning vectors. Cloning vectors are nothing but those are the carriers of our gene. Generally we are trying to insert some gene into something, right? In biotechnology we have to use something as a carrier. We have to use a thing which is having that nature which can help us in carrying this gene from outside to that into inside that genome, in order to incorporate into the genome of the host organism. And that thing, that's something which we are using in order to incorporate or in order to carry this, is called as cloning vectors. These plasmids of these bacteria are generally used as cloning vectors and vectors in this biotechnology. Plasmids are helpful as vectors in biotechnology. Okay, clear? That is about the plasmid and vector. Now the next thing that we require is, yeah, that's it. We require that's it. Yes, cloning vector. About the features of cloning vector also, shall I tell you now? Then the cloning vectors will complete. Yes, let us complete the features of cloning vectors also. Cloning vectors will be completed. Then we will be moving on to the project that is conducted by Stanley Cohen and Herbert Boyer. Then we will be moving on to how this restriction endonuclease is cut, examples of those restriction endonucleases, and we will be seeing PCR technique. Okay, PCR technique and insertional inactivation, all those. Let us see about cloning vectors. Features. Features of cloning VC. VC means vector, not VCT. Okay. Hi. YCT. Okay. ICT. Not YCT. VC means vectors. What are the features that these vectors should contain to be a cloning vector? The first one, it is has to have a region of replication. First of all, I only told ORI site should be present. Okay, ORI site should be present. Without ORI site, it cannot be considered as an cloning vector. And vector without this region of ORI, it cannot be considered as an vector, cloning vector. Next one, selectable marker. What is selectable marker, sir? Selectable marker. These selectable markers are those genes that we are using, that we remove when. Okay. See, for suppose there is a bacteria. There is a bacterial cell. This bacterial cell is resistant to antibiotic. Some antibiotic that is resistant to antibiotic. It is resistant to antibiotic. Now, when this bacteria is resistant to antibiotic, what I am doing is I am using this biotechnology and I am removing this antibiotic resistant gene from that bacterial genome. I removed this antibiotic resistance. Now bacteria is not anymore resistant to antibiotic. It will die. Now the thing is, when this bacteria is put onto that antibiotic resistant medium, it dies. Now which is the selectable marker which is helping us to select between recombinants and non-recombinants? This antibiotic resistant gene, and this antibiotic resistant gene is your selectable marker. Okay, if it should have selectable markers. These selectable markers contain the sites for, it should have different sites for restriction endonucleases, and these should not be repeated. Remember that. Next one, these are only called as cloning sites. These restriction endonuclease sites are only called as cloning sites. Okay. Next one is that's it. These are the three key features in order to consider an vector as a cloning vector. Okay. Actually, in our NCERT there is an example which is given for this vector in order for us to understand this cloning vectors topic very easy. That is a plasmid, one plasmid of this EcoR1. Okay, we'll be discussing about it now. Plasmids of EcoR1. Right? PBR 322, PBR 322. This PBR 322, it is a plasmid. This PBR 322 is a plasmid of E. coli. Okay, PBR 322 is a plasmid of E. coli. E. coli का plasmid. It is a plasmid of E. coli, PBR 322. This plasmid is having all the features that we have said now. Okay, the things which we have discussed now. It is a cloning vector. How? Let me show you its structure now. Now you will only tell me that this, yes sir, this is a cloning vector. This is the worst ever circle that I have drawn in my life. Wait, I don't know how to draw a correct circle in this. Just trying. Yes, this is fine. Oh shit. Let's try now. Now I'm trying to increase the size, guys. That's it. Make it to 46. Now let us try. Yes, this is the circle, not circle. This is your PBR 322. I am changing its color. This is your PBR 322. Now it is having different sites. This PBR 322 gene, okay. This PBR 322 is resistant for tetracycline, so it is having tetracycline resistant gene. It is resistant for ampicillin, ampar region it is present. Now, since it is a cloning vector, it should have a region of replication ORI. It is having another region which is in extra called as a replication of primer. Okay, sorry, repressor of primer, ROP region. And these it is having ORI. First condition is satisfied in the case of which one? Our PBR 322. The first case is satisfied. The second case, it should have selectable markers. It is having these selectable markers. Where are the restriction endonuclease sites? I'll be giving you the restriction endonuclease sites within a minute. These are your selectable markers. Yes, these are your selectable markers. Next one. This is completed. It should have sites of cloning sites. Let me give you the sites of cloning. A trick is there in order to remember. How do I remember these is that only I am telling you. See, in tetracycline resistant gene region, there are two sites for cloning, two cloning sites. Both are of one only, BAM-H1 and SAL1. Both are of 1. Okay, both are 1 in theta region. Both are 1. Next one, coming into ROP region, we are moving like this in anti-clockwise direction. ROP region, you have only one, that is PVU1. Okay, PVU1. In theta region, both are 1s. In ROP region, there is only one, that is only 1. Okay, that is only 1. Next one, AMPAR region. In AMPAR region, you have left here PVU1 only. You have written only PVU1. Sorry, in ROP region you have PVU2. Yeah. Sorry. In ROP region you have PVU2, but in theta region both are 1s. In theta region both are 1s. In ROP region you have only one, that is PVU1. And in AMPAR region you have left here one thing, that is the first one, PVU1. Okay, PVU1. And then the second thing is here PST1. Both in your theta region and AMPAR region you have both ones only, but in your ROP region you have 2, but you have left some others. There are present in the in between of ROP region, in between of AMPAR region and theta region. What are they? HIND3, Hemophilia influenzae D3, CLA1, CLA1, and Escherichia coli R1, ECO R. These are the sites of PBR 322. Now what I do, how these are helping as selectable markers, I am telling you now. This PBR 322, if it is present in the Escherichia coli, it is resistant for tetracycline as well as ampicillin. Okay, now what I am doing is, I am isolating this PBR 322 plasmid from that Escherichia coli, and I am using my restriction endonucleases. What are restriction endonucleases? What they do, they cut in the between, right? I'll be giving an example after this only. Restriction endonuclease is cut in the between, right? So whenever this restriction nuclease has cut in the between of this theta region, I have applied and restriction endonuclease which is suitable in the theta region only. That is BAM-H1. For example, I have applied BAM-H1, restriction nucleus BAM-H1. Now when I have applied this BAM-H1, it cuts this tetracycline resistant gene in the middle. When it has cut this tetracycline resistant gene, what I do is I apply some other gene A, and I have cut this DNA which is isolated from other organism, that gene which has isolated from other organism, using the same restriction endonuclease has to be used. Otherwise same stick ends will not be formed. What are stick ends? Yes, coming, please wait. Same stick ends will not be formed and they will not be link it together. So that is why I have used the gene A, I have introduced here, and I have applied DNA ligase molecular glue. Then this DNA ligase helps in attaching of these two sticky ends. Now when I have applied this molecular glue, what happened? This gene got attached in the region of tectar region, in the tectar region of PBR 322. Now what I did is I have again sent this back into the Escherichia coli and I have left it for replication. Since ORI is present, it is replicate. It is able to replicate and it will produce many copies. Now what I did, unfortunately, I have mixed these recombinants which are not having tectar gene but having gene A with the non recombinants which are usually having, which I have not produced, which I have not done any experiment on them. I have mixed with them, unfortunately. Now how I can select these? How can I select this? There are selectable markers. See on your screens, there are two selectable markers. There are two selectable markers, tetracycline, ampicillin. Now what I did is, I have took ampicillin, antibiotic, ampicillin antibiotic solution. Now what I did is, I have put this all the recombinants and non-recombinants into this ampicillin, or I have poured this ampicillin onto them. Now whenever I have poured this ampicillin onto them, all are alive. Why? I haven't done any modification in the ampar region. I have done it in tectar region using BAM-H1 as a restriction endonuclease in the recombinants. Okay, the task is becoming a little tough. Now what I did is, now I took this tetracycline and I have put this tetracycline onto that bacterial colony which is present with recombinants as well as non-recombinants. When I put, as soon as I put this tetracycline antibiotic, my recombinants die. Why? They are not having the gene for resistance. Tectar gene is cut and the gene A is put. Yeah. Now my recombinants die. Non-recombinants are present. Now like this, I have selected my non-recombinants. I have selected my non-recombinants using the selectable marker tectar gene. Okay. Here tectar has been used as a selectable marker in order to select my non-recombinants. Clear? Clear. Now a little more about the restriction endonucleases, REs. Okay, restriction endonuclease. I have to tell you this first before the plasmid introduction only, but I haven't thought that I would complete the plasmids or cloning vectors and then move on to the experiment done by the Stanley Cohen. What I am doing is, I am explaining all the things, then finally we self will do that experiment which is performed by Stanley Cohen. That would lead to an example also. Yes, how to use all these techniques in biotechnology, you will be getting it. So that is what I thought in between the lecture when I am recording it. Okay, no problem. Now I am talking about ECO R1. ECO R1 has a sites which it is a restriction endonuclease. First of all, endonuclease, it cuts in between the DNA. It cuts in between the DNA, not at the ends. If the restriction enzyme cuts the DNA at the ends, it is called as restriction exonuclease. Here ECO R1, it is a restriction endonuclease. There are some sequences where these restriction endonucleases cut, and those sequences are called as palindromic sequences. What are these palindromic sequences? These palindromic sequences are nothing but, see, let me draw something a little. Yeah, till now we have studied, right? Let me do some comedy with my face. A bindi, okay, like there. Okay, some mustache. Okay, like something. Yeah, and okay, chashma. I don't know how to put this chashma. See that? Some comedy. Laugh for this at least. Okay, no laugh. I'm feeling. Okay, this palindromic sequence. Now come back. I'll be giving you a daily life example for this. Malayalam. Malayalam. What is Malayalam? Malayalam is a language. This is a palindromic sequence. How are you able to see? If you read from left to right, the same word Malayalam comes. If you read from right to left, the same word Malayalam comes. Try Malayalam. Malayalam from left to right. Yes. The same way. Malayalam. Malayalam from right to left. The same. This is a palindrome. This sequence. This is a palindrome. As well as if you are a Telugu student. Vikata Kavi. This is one of the best palindrome that I have seen in the whole world. See, in Telugu, Vikata Kavi from left to right, the same. Vikata Kavi. From the right to left also the same comes. See, Vikata Kavi. If you are a Telugu student, this would work. If you are an English student, if you are an NCERT follower, this would work. Malayalam. Okay. Now give us a palindromic sequence in DNA. You are telling the daily life examples. Okay, wait. Do not scold on me. I'm giving you GAATTC. Take this palindromic sequence. Write its complementary base pairing. Come on, fast. By now you have written this, I hope. Yeah. GAATTC. What is G complement? C. A complement T on the next strand. Okay. T. Again T. Again A. G. C. From left to right. From right to left. C. This is a palindrome. From 5 prime to 3 prime and 3 prime to 5 prime, the sequence repeats itself. This is a palindrome, and this is a recognition site for ECO-R1. And generally restriction nucleases and restriction endonucleases, mainly they cut the sites where these palindromic sequences are there. These ECO-R1 or any restriction endonuclease cuts the site. Cuts the site where it identified these palindromic sequences, and it cuts at a specific base pair between specific base pairs. For example, ECO-R1 cuts between G and A, leaving two ends. See this G here, AA, TTC. Yeah, TTC. The same way here also AA, TTC. No, this way, the other way. AA, TTC. Okay, and G. Clear? It has cut these two. It has cut. Now these two are sticky ends. I'm showing you those sticky ends. Wait a minute. This one and this one. These two are sticky ends. Why I'm calling it as sticky ends? If you put them close, they come on and attach. They both are complementary. Here the hydrogen bonds remain same. Here the 3 hydrogen bonds remain same. They both are complementary to each other, that circle ones. So that is why they are called as sticky ends or staggered ends. So that is why. The ECO-R1 has cut at a site where where there is a palindromic sequence called as GAATTC, and it has cut at a specific base pair between two specific base pairs, G and A, and it has left two sticky ends together exposed, and this sticky ends, this palindromic sequence would repeat many times in the DNA. Wherever this has repeated, the ECO-R1 cuts there. If you put that, the ECO-R1 cuts there. Yes, no. Now there is a big problem. See, okay, understood this about this restriction uniqueness. There is a big problem that whenever this gene, this in this whole DNA genome, in this whole genome, there are many GAATTC sites, and wherever GAATTC site is there, whenever you apply this carbon, it cuts at the place of G and A, G, A, G, A. It divides the whole genome into many pieces, and these pieces are called as fragments, many fragments. But out of these whole fragments, you only require only one fragment. How can you separate that only one fragment that you required? Yeah, then you use gel electrophoresis, agarose gel electrophoresis. Okay, agarose gel electrophoresis. Using this technique, you can identify the fragment that is required for you. Actually, this agarose gel electrophoresis is similar to that of the electrolysis in your chemistry. See, there is a metal plate. Okay, metal plate. 1 minute, let me remove my video, like it is not visible, right? Even then it is not visible, so that I have to move the screen. Okay, for a minute, just after this I'll be again coming onto the screen. See, this is a metal plate, and I put on this agarose gel or agarose gel. It is a conducting gel. Okay, agarose gel is a conducting material. It is obtained from, comment down, it is obtained from an organism only, comment down, it is obtained from a microbe only, comment down. This agarose gel, it is an electricity conducting material. Now whenever I put this agarose gel, I dig pits here. Why are you digging pits, sir? Why to use this agarose gel? Why to dig these pits here? Why to tell us? Yes, these are your pits or wells. These are, as per your NCERT, they are called as wells. Now, these fragments are there, right? These DNA fragments. There are many DNA fragments. There are bunch of DNA fragments. Now, what I did is I gathered them all and I placed them in this wells. And now I place them in this wells. Now, what I do is, remember that DNA is negatively charged. Now, what I do is, I attach, okay, wait a minute. I attach or I connect this to an AC power. This is your power supply, AC, something. Alternating current, guys, not AC, that air conditioner. Okay. Now, the way that you have to connect your positive end and negative end is, where you have dug those pits, you have to put the positive end. The positive end should be there. Sorry, the negative end should be there. If the negative end is there, it becomes as a cathode, and the other ways it should be the other one, the other side, the opposite side should be positive. This becomes anode. Now what happens whenever this electricity is applied? Since DNA is negatively charged, it moves from this pits in towards the anode. This DNA moves like this. While moving, the thinner and smaller fragments move faster towards the anode. Smaller fragments come here, and these larger bada fragments remain backside. The same way happens in all pits. Yes, the same way happens in all pits. This can be visualized like that. See, there is a running race conducted in your school or college. The thin and lean, high or tall fellow runs faster than the fat and stout and barf fellow. Yeah, the same thing is happening here. The smaller fragments are running faster towards the anode when compared to the larger fragments which are left behind. Now you have separated the fragments. You have separated, guys. Separated. Now you have successfully separated them. But how to identify the fragment that is required for me? In order to identify, in order to see them, you should stain the DNA. Okay. You should stain the DNA. Okay. For staining, you use a solution called as ethidium bromide. Wait a minute, the spelling goes wrong for me. This one, ethidium bromide. This is a dye which gives orange appearance when put onto the DNA, this is the dye which gives orange appearance when it is used in this, when it is stained. Okay. Leave, concentrate. See, in order to stain this DNA, I use ethidium bromide solution. This ethidium bromide, when this DNA fragments which are separated are dipped into the ethidium bromide solution and UV light is put on them. UV light, okay, DNA comes here. DNA fragments, UV light, it gives an orange shiny color. It gives orange color, glow to the DNA fragments. When it gives orange color to the DNA fragments, I can simply identify the fragment that is required for me. And I can take that fragment, I can use the restriction under nucleus, I can cut that fragment, and I cut that vector fragment, I attach it using DNA ligase, and I can send that back, that vector, into that host organism, and it replicates. Very clear? Very clear, sir. Super clear, sir. Okay, these are your basic terminologies that we have to see. Now we will do an experiment of Stanley Cohen and Herbert Boyer, guys. Okay, EXPT, experiment of Stanley Cohen and Herbert Boyer. Now we are doing that. Are you ready with me? Yes. Now what they have done is solanum. What solanum? Not solanum. Yes, it is Salmonella typhimurium. Salmonella typhimurium, which causes typhoid. This Salmonella typhimurium, it is a bacteria which causes typhoid. Okay, bacteria which causes typhoid. Now the first thing that I have told you is we have to do the Salmonella typhimurium. It is not antibiotic resistant. Whenever you use antibiotic resistant, this gets died. We have to make it antibiotic resistant. In order to make it antibiotic resistant, what we have to do first? We have to isolate plasmid. So that is what Stanley Cohen and Herbert Boyer did. They isolated the plasmid from Salmonella typhimurium. It is a bacteria. It is having plasmid. And this plasmid is a suitable cloning vector. Yes, suitable cloning vector PCS32. I hope. Okay, hope. I'm very bad at remembering these plasmid names. No problem. Just leave it. Not in our syllabus. Will not be reading. They have isolated this plasmid. What they have did is they have cut this plasmid using restriction under nucleus. They put this restriction under nucleus and they cut this plasmid. Now this plasmid was this. They cut this plasmid into this. Now they bought some antibiotic resistant gene, and they cut this gene. They have isolated. How they have did the fragments? They cut the DNA first using the same restriction nucleus. Okay, I am naming it as 1. Restriction nucleus 1. The genome they have cut from some other organism. They have isolated this DNA. They have cut this DNA using the same restriction under nucleus 1, which they cut the plasmid. Okay, why I have told already. Now after cutting it, fragments are obtained. After fragments are obtained, they did gel electrophoresis. They chose the fragment which is having this antibiotic resistance, and they bought it and they put this here, and they used DNA ligase molecular glue and they attach it to this plasmid. Now this plasmid has become antibiotic resistant. Whenever this plasmid is introduced into Solanum typhimurium, since it is having region of replication, it forms many copies, and this Solanum typhimurium eventually becomes antibiotic resistant. This Solanum typhimurium eventually becomes antibiotic resistant. Got it? Very clear? Yes, we have did the experiment that is done by Stanley Cohen and Herbert Boyer using our knowledge which we have done. Yes, I have not gone that deep into that basis level from isolating. Now we will be doing PCR, polymerase chain reaction. Okay, this is also very interesting, guys. Listen, this is also very interesting. Please do not move away. I'll be just taking next other 15 minutes of your time. Okay. PCR. See, PCR, polymerase chain reaction. This is also very interesting. In this, there are different steps to be followed. First one, isolation of DNA. You have to isolate the genome. Isolation of genome. Okay. I haven't told you where this PCR is used, right? Yeah. This PCR is generally used as an amplification technique. It is an amplification technique. It is used to amplify a single gene into many genes. Okay, it is used to amplify that. PCR, not PRC. What happened to us today? Okay, PCR. Polymerase chain reaction. There are different techniques that we have to do here. First one, isolation of genome. Second one, cutting. Okay, cutting at specific location. Third one, amplification. Yeah, have you ever seen amplifier? The sound amplifier, your speaker box. What it is? The minute sound. This minute sound, it amplifies like this. It amplifies very much, and it produces much sound. Okay, amplifier, amplification. Next one, insertion of recombinant DNA. After amplification, we introduce the gene which is required recombinant production. After that, we finally obtain the gene product. Okay. Now, starting with the isolation of genome. I have told you this cutting, I'll not touch that part. Okay, cutting I'll not touch. Amplification I'll be telling you, since I have to tell you about denaturation, annealing and extension steps. Recombinant I'll be telling you how to introduce, how this insert is introduced into the cells, but I'll not be telling you again that genome pasting, all that. No. Next one, gene product, nothing but much are produced. That's it. I'll be not again talking about this gene product. Now, first of all, coming into isolation of genome. This technique of isolation of genome is called as spooling. Not spooning, but spooling. Okay, spooling. What is done is the DNA or genetic material is isolated, okay, or extracted. Okay. The DNA is extracted. After it is extracted, it is dipped into chilled ethanol. When it has been dipped into this chilled ethanol, what happens is, it is an alcohol. What happens is, small thread-like structures. See that diagram in the NCERT which is shown. See this. There are small thread-like structures of DNA. Much thread-like structures like this are obtained when chilled ethanol is applied. Now you use an instrument called as a spool and remove this DNA. Spool is like this somewhat. You use this spool and take out that DNA. This is called as spooling or isolation of genetic material. In order to extract this DNA, what you have to do is, the DNA is present inside cell. Where inside cell? Inside the nucleus. In order to go into that nucleus, what you have to do? You have to first eradicate this cell wall. If you are using a plant cell, bacterial cell, or a fungal cell, in order to eradicate this cell wall, you use a cellulase, an enzyme in plant cells, in order to eradicate a cell wall. Okay, plants, okay, not cell wall, plants, right? Plants, cellulase in plants. I'm putting P, plants. This cellulase is an enzyme that digests cellulose, which is a material of plant cell wall. Or you have to use chitinase if you are using fungal cells. You have to use chitinase if you are using fungal cells, or you have to use lysosome if you are using bacterial cells. What these three do is they cut this cell wall. After the cell wall is cut, only cell with a cell membrane and nucleus is there. Now inside that nucleus only not DNA is present, RNA is present, DNA is present, proteins are present, lipids are present, etc., etc. Many are present. Sorry. Okay, you have to only, you want that DNA only, you want this DNA only, not RNA or all those. This DNA, what you do is you put RNase. No, RNase is an enzyme that digests RNA. Now RNA is gone, you remove proteins and lipids, now all are gone, deleted, only DNA bach gaya. After, when this DNA is extracted, this is extraction of DNA. When this DNA is extracted, it is dipped into chilled ethanol. Some thread-like structures are formed using a tool called spool that is removed out. This is your isolation of genetic material. Now cutting of genetic material, no need to tell. Let's remove that. No. Amplification. Coming into amplification, you have to amplify this small DNA fragment into many DNA fragments, and that technique is only PCR. What you do is you do polymerase chain reaction. Actually, PCR, in this reaction, multiple copies of DNA of interest are synthesized in vitro using two sets of primers. Okay, small chemically synthesized oligonucleotides that are complementary to the regions of DNA, and those are your primers actually. Okay, no, and the enzyme DNA polymerase and primers, DNA primers are used in this reactions. Okay, polymerase in polymerase chain reaction. Okay, next one. Now next one, what you do is you heat this DNA, you heat it. When it has been taken to higher temperatures, the hydrogen bonds break, and two strands get separated. Yes, two strands get separated when you supply heat to it. When two strands are separated, you put this you use a Taq polymerase, which is an which is obtained from a bacterium called as Thermus aquaticus, which is resistant to higher temperatures. Even higher temperatures it is resistant. You use Taq polymerase and you do extension. Now these two strands are producing their own strands. They produce their own strands. They complementary base pairings. Two new strands are formed. Again you put this Taq polymerase. Again this new strands are formed, and again this is repeated many a times in order to produce many chains of the desired character. Okay, next one is your insertion. How can you insert this into the plasmid that you want? Actually, there are different techniques like a gene gun method, micro injection, gene gun method, and pathogen method. These are the various methods through which you can insert this gene. What is gene gun method? If there is cell wall surrounding the cell, especially in plant cells, it is very hard. Cell wall is very hard. What you do is you use biolistics. These biolistics are generally made up of tungsten or platinum. Okay, tungsten. Biolistics are small bullets which are made of tungsten, and this DNA, these biolistics are dipped into this DNA, and using a gun, they are shot into the cell. Whenever they are shot into the cell, through this cell wall, they penetrate deep into the cell, and they go into the cell, and they leave that DNA there into the nucleus. Next one is microinjection. Microinjection is used when there is no cell wall, there is only cell membrane, and this microinjection is an injection which is given directly into the nucleus. DNA is injected directly into the nucleus. Next one is pathogen. We have different pathogens, if you take an pathogen of plant which is used for these scenarios is your Agrobacterium tumefaciens, TI plasmid. So this, using this plasmid, this causes diseases in plants. Tobacco help, yes. In tobacco it causes diseases. Using this you can use it, or you can use retroviruses or retrovectors for animal cells. Okay, these are the types how you insert your DNA. Now there is one final topic that is left, called as insertional inactivation. What is insertional inactivation? Now I will not use this, just listen. Now what we have done is, in the previous one, we have cut the tetra, right, in the PBR 322 part here. Where is this? One here, yeah, here. We have cut this PBR 322 using BAM H1, right? BAM H1. Now we have inserted another gene A. Yes. Now what is happening here is, this tetra gene is inactivated due to the insertion of the other gene A. Okay, it has been inactivated. This tetracycline resistant gene is inactivated due to the introduction of a gene A. So that is why it is not resistant anymore to tetracycline. That is why these recombinants die when they are put into tetracycline antibiotic solution. Okay, so that is called as insertional inactivation. So that is it, guys. I have tried to cover most of the NCERT. If at all something is left, you give one reading to it, and that is it. I'll be meeting you in the next session with another one shot lecture. Okay, with another one shot. Okay, this is Vamsi signing off, guys, from Biolab India, and I hope you have enjoyed the session. If you have, please hit that like button very hard, and also subscribe to our channel Biolab India. And yeah, have a great day further. Jai Hind, Jai Bharat. Bye, guys. Thank you for watching my video.